Enzymes and digestion. Inside your body, thousands of chemical reactions are happening all the time. These reactions are responsible for everything from releasing energy to building new cells. However, these reactions must be carefully controlled. If they happen too slowly, your body cannot function properly. If they happen too fast or in the wrong way, they can damage cells. One simple way to make chemical reactions faster is to increase temperature. But this is not a good solution for living organisms because high temperatures can damage cells and proteins. This is why the body uses enzymes. Enzymes are biological catalysts. This means they speed up chemical reactions without being used up or changed in the process. They allow reactions to happen quickly at normal body temperature. Enzymes are proteins. Each enzyme has a specific shape that is essential for its function. This shape includes a region called the active site. The active site is where the reaction happens. Each enzyme only works with one specific substance. This substance is called the substrate. The shape of the substrate must match the shape of the active site. If it does not fit, the reaction will not happen. This idea is often explained using the lock and key model. The enzyme is the lock, and the substrate is the key. Only the correct key fits into the lock. How temperature affects enzymes. Temperature has a strong effect on enzyme activity. At low temperatures, particles move slowly. This means there are fewer collisions between enzymes and substrates, so the reaction rate is slow. As temperature increases, particles move faster. This leads to more collisions and a faster reaction rate. However, if the temperature becomes too high, the enzyme loses its shape. The active site changes and the substrate can no longer fit. This is called denaturation. In the human body, most enzymes work best at around 37 degrees Celsius. This is called the optimum temperature. How pH affects enzymes. The pH of the environment also affects enzymes. If the pH is too acidic or too alkaline, the structure of the enzyme changes. This again affects the shape of the active site and can stop the enzyme from working. Each enzyme has its own optimum pH. Some enzymes work best in acidic conditions, while others work best in neutral or alkaline conditions. For example, the enzyme pepsin works in the stomach. The stomach is acidic, so pepsin works best in low pH conditions. Investigating enzyme activity. Scientists often investigate how enzymes work by measuring how fast a reaction happens. For example, the enzyme amylase breaks down starch into sugar. To test this, starch is mixed with amylase and samples are taken over time. Iodine solution is used to test for starch. When starch is present, iodine turns dark blue. When starch is no longer present, the iodine stays orange. The time taken for the starch to disappear tells us how fast the enzyme is working. The rate of reaction can be calculated using this idea. Rate equals 1000 divided by time. This means the shorter the time, the faster the reaction. Digestion and enzymes. Digestion is the process of breaking down large food molecules into smaller ones. Large molecules, such as starch, proteins and fats, cannot be absorbed into the bloodstream. They are too big. Enzymes break these large molecules into smaller, soluble molecules that can be absorbed. There are three main types of digestive enzymes. Carbohydrases break down carbohydrates into simple sugars. Amylase is a carbohydrase. It breaks down starch into sugars. It is produced in the salivary glands, pancreas, and small intestine. Proteases break down proteins into amino acids. They are produced in the stomach, pancreas, and small intestine. Lipases break down fats into fatty acids and glycerol. They are produced in the pancreas and small intestine. The role of bile. Bile is produced by the liver and stored in the gallbladder. It is released into the small intestine. Bile has two important functions. First, it neutralizes the acid from the stomach. This creates the right pH for enzymes in the small intestine to work. Second, it breaks large fat droplets into smaller ones. This increases the surface area for lipase to work on, which speeds up digestion. Food tests. Scientists can test foods to find out what nutrients they contain. To test for sugars, Benedict solution is used. If sugar is present, the solution changes from blue to green, yellow, or red. To test for starch, iodine is used. If starch is present, the color changes to blue-black. To test for proteins, the Biuret test is used. If protein is present, the solution turns purple. To test for lipids, Sudan solution is used. If lipids are present, a red layer forms. Question and answer section. Question: what type of biological molecule is an enzyme? An enzyme is a protein. Question: what is the function of an enzyme? An enzyme speeds up chemical reactions in the body. Question: why does increasing temperature increase enzyme activity at first? Because particles move faster and collide more often. Question: why does a very high temperature stop enzyme activity? Because the enzyme becomes denatured and its active site changes shape. Question: why can enzymes only catalyze one reaction? Because the shape of the active site only fits one specific substrate. Question: which reagent is used to test for glucose? Benedict's solution. Question: what does it mean if there is no color change in Benedict's test? It means there is no reducing sugar present. Question: which reagent is used to test for starch? Iodine solution. Question: what does a positive iodine test look like? The solution turns blue-black. Question: what does pepsin need to work properly? An acidic environment. Question: what do the results of the pepsin experiment show? Pepsin works best in acidic conditions and does not work well without acid.